Testing, testing. Okay. Mm.
Sorry about the delay.
Okay, wait a minute. Yeah, okay. Can you hear me? Can you hear me?
Yeah, I can hear you.
Oh, okay, okay, cool. All right, cool. I was wondering if I had a audio issue. All right, so let's see. All right, so basically, um, I guess what I want to establish is the fact that fusion did not happen, and I want to base that on the fact is that it's highly unlikely. I want to base that on the fact that that type of fusion never been recorded in history, and I'm referring to a telomere to telomere fusion. Um, and I also want to make a point that um, those type of fusions are not necessarily safe, and it's also the thing that when you have a fusion like that, that first human that diverged from the common ancestor would have to find um, another human with the same type of the same type of chromosomal arrangement and it's on the same locations. Do you agree with that? In order for it to have viable offsprings.
Well, no, not really. I mean, what you need is for the offspring to have the same genetic complement.
Right. Okay. So they need to have the same the same fusion on the same two chromosomes. Okay.
No, they don't. So if you have two chromosomes A and B, right, that fuse, um, and we'll just say that it's in the fusion happens in the dad, then just a second.
Sure.
Sorry. Then then obviously from the mom where there's no fusion, the offspring is going to get A and B, one copy, right? From the
But it,
dad, hold on. So from the dad, they can either get depending on how the chromosomes separate, they can either get uh, basically the fused copy with one of the others, right? In which case they've got more than they should, or they can basically not get anything, less than they should, which is also bad. But if they get the fused copy and nothing else, with the unfused from the other parent, then they have the normal genetic complement, which is perfectly viable.
No, but then they would have to meet, another offspring, if they want to mate with someone else, they're going to have to meet someone with those same, the same arrangement in the same location. Otherwise, you're going to get some kind of imbalance. You're going to have issues there, because I can't see how you're going to start a new species off of a defect, and that is a defect. I mean, it's not normal to have a fused chromosome.
So there's actually a couple of families that are living with a chromosomal fusion event, and they actually do propagate this through several generations, and the viability for the offspring is lower than what you'd expect, because of this assortment issue that I was trying to describe. But they do produce viable offspring, and when the offspring are viable, they're perfectly healthy. I mean, they're normal. It's just that, you know, it's a little bit more difficult for them to have children, but the children that they have are fine.
Okay, but the thing about it is, so basically what's happening is 46 chromosomes would have to be the dominant chromosomes. I mean, everybody pretty much would have to basically find a way to have 46 chromosomes, you know, because if you got 48 and you got one or two people, a few people that have 46, then 46 has to be the dominant one. Okay, that's unlikely. That means if that's the case, well, how come we all don't have more people that has unbalanced chromosomes right now, if that's the case? So what I'm saying to you, it's unlikely that you're going to go from 48 chromosomes and then 46 chromosomes is going to be the dominant species. That's what I'm saying. And you do have
well, yeah, but I think that at the outset you said you were going to try to prove that it was impossible, not.
Okay. So, I tell you what. Let's do this then. Let's say highly, highly unlikely. Okay. Let's say highly, highly unlikely. And I plan to build this case because.
Yeah, sure, but I mean, so like, if you say that it's highly unlikely, but you have evidence that it happened, then.
Okay. Well, I mean,
something unlikely happened. That's
right. Well, yeah. I mean, you really don't. I think it really comes down to how you view the evidence, how you look at the evidence. But like what I'm saying to you is, and when I say highly unlikely, I want to really, really stress that, because like I said, you know, you got 46 chromosomes, or you got unbalanced chromosomes, and everyone else has 48. You know, it doesn't seem like that's going to be the dominant species. But I don't want to stay too long. I'll let you finish with that because I don't have a lot of time. Okay, are you there? You got two things going on here.
Yeah, sorry. I was just doing that because it's easier for me to screen share from my other computer.
Okay. Okay. So let's not spend a lot of time on that. We both kind of agree that it's, at least we're going to say this, unlikely. Okay. Let's move on from there. Okay. So.
Can I just show a quick diagram of that assortment thing I was talking about?
Yeah, I actually seen those. I've seen that before. The people, there was different. One in China, one was in, I don't know if it was Switzerland, something like that. I know how that works with the rearrangements, but you can show it to me anyway.
Yeah. So you can
tell me when you're ready for me to look at that. Oh, I can
go ahead.
I got to click onto it. Okay, I got you. Yeah, I've seen this already many times.
Yeah. So what this is showing is you've got the fusion event here. And you can either get assortments where you have normal assortments. So these are the unfused chromosomes. Normal complement with a fused chromosome. Abnormal complement, abnormal complement, abnormal, right? So basically, 2 of these out of the 6 possibilities, 2 of them have a normal complement of genetic material. The only option from the other side is a normal complement, right? So either of these 2 options are viable. The others are not viable.
Right.
But so in other words, so this is how you start getting it into the population.
Right.
Is it starts spreading like this, and these are viable, and then eventually, 2 of these meet up, right? And now you've got somebody who's got 2 sets of the same fusion.
Yeah, but but still they have to be in the same location too, though. See, that's the thing. Those
You mean geographical location, right?
well, I mean, what I mean by say location, I mean is that if you have a fusion of let's say 14 and with that 14 and 16 there, those same ones got to match up with the okay. So
Well, no, yeah. So right, no, that's
Right.
obviously true. But what I'm saying is,
But-
you have a single initial fusion event, right? It's this initial fusion event that gets propagated throughout the population. Okay, so it's only this one fusion event between
Right.
14 and 15 that it shows, and then so it gets propagated throughout the population, and then 2 people carrying this meet up, right? They have offspring, and some of those offspring are going to have, only have the fused chromosomes. So that's what I'm saying. Now,
Okay.
obviously, I don't.
Okay, I respect that. I respect that. But still, you have something that's not very viable, and still you have to make 46 chromosomes the dominant species. Okay, so let's move on to the next thing, because I'm building. My case is extremely highly unlikely. Okay, so I guess the next step would be, they had you say the fusion event happened basically, and I guess I want to get into the fact is that what we're looking at is the first documented telomere to telomere fusion. Would you agree with that? That didn't involve cancer. That's not involved with cancer lines. Do you agree?
So I'm not sure if it's the first necessarily. It might have been the first, but it's certainly not the only. There are a number of other documented fusion events, telomere to telomere fusion events in pigs, in antelope, in rats, in zebras.
Okay, now, well, hold on for a minute. Hold
several.
on. Are we talking about telomere to telomere? Or are we talking about?
Yes.
Okay, well, see, I ain't really come across that yet. I come across satellite DNA to telomeres fusion, but I haven't seen that one yet. Telomere to telomere, because every time when you see even the authors who basically came up with this, they wasn't looking for telomere to telomere, because really, honestly, telomeres are basically machines that's made not to fuse. That's the purpose. So that's the reason why it's highly unlikely.
The authors who initially postulated the telomere to telomere fusion event on between the human chromosomes were doing it based on G banding comparisons.
Okay.
And if you look at the G banding comparisons, it's pretty obvious that it's a telomere to telomere fusion. So when you actually then go to do the sequence work, that's the only thing you would expect to find, and it is actually what you find.
Well, yeah, you saw some banding. You saw some banding that looked similar. Fine. Okay, but see, the thing about it is, you have chimp DNA and human DNA is pretty similar anyway. So you're going to see a lot of similarities anyway. Do you?
Yeah, it's extremely similar.
Okay,
it's 90% or so.
I'm not going to say I don't really want to get off topic with that, but I'm not going to say it's in the 90s, because I'm going to be real brief with that, because of the fact is there are lots of DNA that's present in chimp that's not present in human, and vice versa. And there are unalignments that doesn't line up. And as a matter of
2% or so.
No, no,
2 or 3.
it's actually more than that. It's actually more than that. And then you can't get people to agree which genome is larger. So it's not something that you can just like anybody really have a really good answer for. It's definitely up for debate. Do you agree with that?
Not
Or you gonna stick by? Or you
particularly.
gonna stick by the 2% because the 2% fits your cause as far as bringing it up to 90%.
No, I'm gonna stick with that because that seems to be consistent with the evidence that I've seen.
Okay, all right. Well, I'm not gonna I'm not gonna like overly argue about stuff. Okay, so so you got these banding patterns, and these banding patterns are homologous, and so we want to assume that since those banding patterns are homologous, that there was a fusion that took place there, with in a situation where you have telomeres that has short-term protein and protectors and telomerase, so they do not fuse. But then you got a fusion here. Okay, so now if we look at if we look at those actual chromosomes, if we look at that fusion site there, that fusion site to me, the way I look at it, that fusion site to me, you had a few guys who looked at to see, it was making a comparison to see what type of DNA was on there, and if you look at that whole region, that 798 kilobyte region, that's right there in that fusion area. There's no chimp DNA in that region at all. There is a gene that spans the fusion site, the DDX 11L2, but there is some a family of DDX 11 genes is there, but out of that family, out of 18 copies, you have no chimp DNA there. As a matter of fact, out of those 18 copies, you only have 2 copies that is chimp and 4 that is gorilla, but they're not found on those chromosomes.
Yeah. So the interesting. Hold on a second. Is that echo from my end or yours?
I don't know, sir.
I'll see if it fixed. Okay, I think that fixed it. Okay. So the interesting thing about the DDX11 family is that it seems as though they're only found in near telomeres, with the exception of DDX11L2. So
I disagree with that.
Okay, so where there's a DDX that's not by a telomere?
Well, see, the thing about it is I think that what you're saying, okay, first you got telomeres, you got subtelomeres. Okay, so, okay, tell you what, make your point first, and then we'll go from there.
Well, my point is just that you've got a dozen or so more, I think, of these DDX regions, and they're all found in subtelomeric regions, found by telomeres, that is, except for DDX11L2, which is found by something that looks like a fusion site, and so that actually tends to support the idea that it's a fusion event, not go against it.
No, well, but the problem is you're missing a lot of in this fusion area. You're missing a lot of chimp DNA that should have been there. You should have had chimp DNA there. As a matter of fact,
well,
not
you wouldn't.
Hold on one second. Not only are you missing chimp DNA there, and you would have saw some chimp DNA there in that area, because there are significant amounts of chimp DNA on the 2B chromosome in that same area on the chimp that was renamed. So you don't have no chimp DNA in that area. Okay, you have a significant amount of chimp DNA in that area on the chimp 2B. Okay, not only that, you're missing about 24 megabytes of information there, period. Okay. Now, why are you missing that information if the telomeres fuse, but the fusion stop in the subtelomeres? You shouldn't be missing DNA there.
And just 1 second, please. I want to try to get
Sure.
something up here.
Okay. And I do want to say, I do want to say to you that what I'm telling you is absolutely correct, because they do have
Okay.
rescue missions for this. No,
Can you see this? What I'm sharing here?
Hold on, let me click over to that and look at that. Okay, I can see that.
Okay, so this is a Sentinel map based on genotyping data, and what you can see here is the Sentinel with human chromosomes in the middle, and mouse genome is on the left side, and chimpanzee is on the right, and it's colored based on the chromosome that it matches to. So if I just scroll down, see that it breaks between mouse and then again, and you see how this is all in order though on the chimpanzee, then breaks again on the mouse, and again here and here, and then here's the break.
Okay, so the mouse is on the left hand side there.
Yeah, it's, and then so here's the break, the color change between human and chimpanzee. And this is where it jumps from 2A to 2B. And then if I keep scrolling down, you see it's all in the same order still.
Okay.
All in the same order, all in the same order. So, and here's where you get that area we're talking about. There's DNA in chimpanzee, not in human. There's a little bit of a insert there.
There's a lot of DNA that's not. Well, you know, I don't really know how that have any real significance. To me, that's kind of off topic.
No, it's perfectly on topic, because what I'm showing here is that you have on human chromosome 2, you have the entirety of the chimpanzee of the gray chromosome 2A 2B on one side or the other, with very, very minor little differences. When you're talking about these inserted regions, they're very small with respect to the overall size. I mean, if you talk about, because this is a 240 megabase region, the chromosome. So if you talk about, you know, a couple hundred kilobase or something, it's really not that much. So
okay, but see, that's your evidence. But my evidence says that DNA, the DDX DNA, I'm sorry, the family, the helicase family. My evidence is saying that there's only two, okay, in that region, in that area, and and on that particular chromosome, there's only two copies of it, okay, and that's right there in the area. And on that chimp chromosome, there is specific chimp DNA chromosome specific to chimp on that 2B. It's not present in human. So, I mean, to me, if you want to, you can look at your evidence and you can look at that right there. But this evidence is documented. I don't know how to screen share, but hold on, let me see. And then, like I said, you're missing a gang of DNA. You're missing a gang of DNA there. Now, you can't be missing DNA if the telomeres did its job, not a good job, but it did stop the damage from coming to the telomeres.
Sorry, wait, you're saying that there's DNA missing from the subtelomeric region, or from inside the.
It's missing. It's missing right over there next to. Now it's not just the telomere, subtelomeres. It's missing right there on the. It's missing the chimp DNA. That's just not specific to the subtelomere. It's missing a lot of DNA there. A lot of.
And it's missing. It's present on the chimpanzee, not on the human, or the other way around.
Let me see if I can. How do you screen share?
You see the little the green icon on the left, second one from the top.
Right there. Okay. So now I have to, so now I have to click onto the window that I'm talking about.
Yeah, it's best if you select the specific window instead of share the whole thing.
Okay, yeah. That's what I'm gonna do. I'm gonna pick the whole. Let me. Okay, wait. Let me get off screen share for a second. Okay, and that way I could find the exact one I'm talking about first.
Yeah, because if you just pick one window, then it's a cleaner presentation than if you just show your whole screen. It causes problems.
Right. And the thing about it is, this information is okay. Is that one right there? This information is. I mean, this information comes from a lot of Okay, what happened there? Did it pop up there or not? Oh, I didn't do it yet. Okay, wait a minute. Okay, so I hit screen share. Okay, then I hit share. Is that what I do? No, it said no. How about application window? Okay, that's what you do, right? And okay, and then go to the application window. Is that what I do? And then I go to the page.
Just let me follow along here. It's
Okay, wait a minute.
application window, and then you pick the one that you actually want to show, and then you hit share, and then it should.
Okay, wait a minute. Hold on. Let's see what we got here. Oh, then hit share. Okay. Okay, so is that it right there? Okay, fine. That's it right there. Okay, so let me see. I can scroll this up or not. I don't know how that works. Yeah. Okay. Do I have to like scroll on my page like this? Is you seeing what I'm doing?
Yeah, I can see what you're doing.
Okay, you have to do that from there. Okay, so yeah, this is what it's saying right here, and this guy's Ventura. Okay, first you had Unic and Partridge in 1982. Basically said that you had 24 megabytes of DNA missing from the fusion. So that's a lot. And then it says, and then you also have right here, it says recent analysis characterizing the DNA sequence and genomic organizations of chromosome telomere and cap regions of gorillas and chimpanzee. Researchers found a large amount of chimpanzee specific satellite DNA at the ends of chromosome 2B, but not present in chimp. Ventura 2012. The authors, and this is the escape mission. This is why we know it's real. The authors postulated that the chimp satellite was deleted at the fusion event. See, that's the rescue mission. Okay, but the thing about it is, that's a lot of DNA to be rescued. So you need a rescue mission for that. Okay, to say what happened to all that DNA. So you got a lot of chimp DNA that's missing. You got hold on.
Yeah, is this?
Okay, hold on, just one second, and then you got and then you're saying that you're saying that the mutation rate for the damage of the telomeres, okay, was way above what they would expect to happen within a 6 million year time span. I'm gonna let you take it from there.
Yeah, so this is the subtelomeric region. I think in between the on the short arm of the one side of the fusion, right? So usually you don't see much in the way of coding functionality on those anyway. So
wait, hold on. What do you mean? You don't see?
So the most common.
Okay.
So the most common.
Okay. So are you saying you don't see long coding RNA genes there? You don't see long coding RNA genes there?
What I'm saying is that for instance, the most common form of translocation is the Robertsonian, right? And the balanced Robertsonian translocation, you end up getting the two short arms fused together, and those are usually just lost. Okay, and so basically, what they're suggesting happened here is that in the head-to-head fusion, the short arm that's between the centromere and the fusion site is lost. Okay. Now that's just like if you lose the short arms and a Robertson fused together in a Robertsonian translocation, which usually doesn't do anything to an organism. So what I'm saying is there's usually not much of any great
Okay,
significance in those regions.
well, but you got DNA that's missing there. But see, so here's the thing. Here's the thing. Now, first of all, see what I'm saying. I'm building this case against it. First, we want to deal with the fact is that it's very unlikely and rare that 46 chromosome humans would take over. Okay, it's also very unlikely that with the known mutation rates, is very unlikely that over 6 million years maximum, and it could be 3 million years, that those telomeres would have depleted to that amount. Okay. Matter of fact, Fan was astonished anyway.
Depleted to what amount?
Okay, if you had a fusion, a telomere to telomere fusion, you know, the telomeres would typically be between 5,000 to 15,000 telomere sequences on the end of those chromosomes, correct?
No. If you have a fusion event, your
No.
telomeres are already short.
I understand that. So what happened is you had a lot of fusion that happened because it got all the way down to like 800. So you had a whole bunch. So that means you had a lot of mutation. Okay. You follow?
No.
Okay. So how do you get
No.
down? How do you get down? Okay. So how do you get down from a possible 30,000 down to 800 then? If you didn't have a lot of mutation that damaged those telomeres.
So you have telomere shortening, right? It's just sort of a natural process, and this does not.
Telomeres, but you got telomerase. You got telomerase that adds telomeres on the end of that. So
Yes,
it
okay, should.
So and that comes with age. Okay, so unless this human got extremely old. Okay.
Yeah, that's one way, but you also have various other processes that can occur that can cause shortening of telomeres, or just loss of certain regions of telomeres. It's not only the one thing, and it's also important to realize that the shortening is not necessarily uniform, and the repair is not necessarily uniform.
Well, I mean,
so you can get these.
What? Okay. I'm sorry. Go ahead.
Yeah. So just that you can get these variations in the lengths of the telomeres.
Yeah, but
so
go ahead. I'm sorry. I'm sorry. I thought you was John. Go ahead. I'm sorry.
Yeah. So that's how you get down to the shortened length to allow for the fusion event in the first place, right? But once the fusion event happens, you're not going to lose anything from within that site at any rapid rate.
Okay. Well, the thing is this here. Okay. So this is what we're dealing with. We're dealing with you telling me that some other method, some other mechanisms decrease those telomeres to that degree when telomeres are built with proteins on the end in their machines that basically keep the overall chromosome stability together. So you got these machines that never fuse in history. Okay.
Well, again, they
hold on.
There are fusion.
Well, I mean, okay. Well, okay. Well, I'm gonna bend a little bit because I feel like I can. Extremely rarely. Okay. I'm gonna bend a little because I feel like I can. Okay. Very rarely fuse because they're pretty much a safeguard. So even if we want to say that it happened, they pretty reliable. Okay. So what you have here is you have these telomerase wasn't working. Okay. It didn't add the telomere to the end of it. Okay. You got down from a possible 30,000 down to 800. Okay. Then what you're saying is it stopped. So we didn't get and I'm pretty sure that was Isaiah and it was also it was Ruth who said that it stopped right there in the subtelomere region because we know what we saw the TDA GGG, which is degenerate, but they were still there. So that means that the damage stopped in the subtelomere region. Okay, so We
Well,
got
in the telomere itself, not the subtelomere.
okay, the telomere itself, fine. But you got to fight with Wolf on that one. So basically, we got lost genes there. A lot of lost genes, a lot of lost DNA information there. A lot of it missing from chimp to human. Okay, and like I said, well, I didn't show you that yet. I'm sorry. Let me see. Here we go. When they was doing it, and this is what Ventura. This is you know, they basically said that they probably lost that through they lost that through, what did he say he lost that through the it got deleted. Authors postulated the chimp DNA was deleted in the fusion event, yet the telomere sequence distal to it somehow remained. That means that the telomere was still cool, but we lost that information some kind of way. Okay, that's kind of hard to swallow. Okay, so now you got Fan talking about he put another a serious problems here. Again, it was talking about the if you can still am I still screen sharing here? Hello?
Yeah, I still see it.
Okay, so you had a lack of synteny of gene content on the DNA sequence similarity on the chimp compared to what you were seeing. You losing a whole bunch of chimp DNA right there. And then
Well, I mean, again, the synteny is extremely good across the entirety of the chromosome. So I mean, arguing that it's not very good in some tiny region is not terribly impressive.
Yeah, but you got a huge region here that's and then you have but then you have DNA, then you have this DDX gene, this DDXL2 gene that's not even present in chimp at all, that spans the fusion site. So it's kind of hard to believe that you can slam a couple of chromosomes together.
This is actually something really important, though. The DDX11L2, the canonical transcript does not span the fusion site. There is a putative transcript that spans the fusion site, but if you actually see the expression data for that, it is to say that it's not expressed.
Okay, the thing about it is, it depends on where you look. It depends on where you look, partner. If you look at different data sets that collect information, different ways of collecting the data. For one thing, let me just say one thing. The way we collect data as far as gene expression sometimes is pretty sloppy. Okay. And then you got different type of data sets that are better at certain things than other ones are. Okay, so you have this one I showed them. Let me see this site. Gosh, wait a minute, where we at right here? Oh, that's us. Wait a minute, hold on. Where we? Oh, okay, wait a minute. Oh, I'm just messing this all up. Okay, hold on for a minute. I didn't mean to do that. Wait a minute. Am I back to one screen? Am I back to one thing sharing? Or
yeah,
not?
you are.
Okay. So you have this site called Harmonize. Hold on, let me see. And they basically saying this right here. They saying that this gene right here, this DDX LL2 gene, is regulatory RNA helicase, which encodes several transcript variants, because it actually has a short one, a long one, and actually that fusion has been proven to be in that first intron. Okay, that first intron, it's not really a fusion, it's a promoter. It's the second promoter in the first intron. And it says that is expressed in 255 different cell types and tissues. Specifically,
that's not true.
Well, I mean, that's what you say, but it's like,
well, I mean, he's just categorically wrong.
Okay, well, let me hold on for a second. Let me try to go over to an email. I probably should stop screen sharing for this right here. He also
that would be a good idea. Yeah, you're gonna want to you do want to stop your screen share.
Okay, hold on.
Just hit the green button again.
Hit. Hit the what button? Oh, green button again.
The green icon on the side. Yeah, there you go.
Cool. Okay, so let me go back over. Let me go over to. So one thing there was this one site. Let me see if I can find it real quickly. Ace right here. Well, I'm not screen sharing here. Ace said that this gene is moderately expressed. This is Ace view. Do you want to see that? Are you with?
Sure.
All right, hold on for a second. Let me see if I could. I need to get some of this stuff closed here. Okay. But what I'm saying to you is, it's very sloppy how they express genes. A lot, for one thing, that DDXL2 gene is not present in mouse or chimp. So really what you can't do is, you really can't even test that, because you got to knock that gene out. You can't test that gene in mouse. You can't test that gene in chimp to really know how highly expressed this gene is anyway.
Well, you can tell how
You have to
expressed it
do,
is, or
you have
not
to
in
do
human though.
exactly, but it's not in human, but it's not in nothing else though. It's specific to human. That's the
Yeah,
problem.
so that's not evidence. That's not evidence of great functionality.
But that's not evidence of non-functionality either.
Well, that's true, but I mean, if you're going to talk about something that's only found in humans and it's not expressed at any great level in humans,
Okay, are
then
you?
that's not.
Yeah, but that's what you say. Okay, so right here, I'm looking at. Can you see what I'm looking at? Am I screen sharing here?
Yeah, I can see
Okay,
it.
so right here it says right here, according to this gene, is moderately expressed.
And where did they get their data from?
I don't know. Uh,
Can you click on that?
Moderately expressed. What you mean? What you want
Yeah,
me to?
yeah, yeah. Click on that. Click on moderate. Yeah.
And then another thing is, then you and then see the thing is you have this DDX gene, which they say is moderately expressed, but this other site says it's highly expressed, but then it's also highly co-expressed with.
Yeah, co-expression is
Right.
not an
Okay.
accurate stand-in for anything. Hey,
See, that's,
can you present me for a second though? I just wanted to, while we're talking about this, I just want to
okay.
show
Okay.
something.
Hold on. Let me. Yeah. But okay. But let me talk while I'm presenting you. See, and see, that's the whole. Oh no! Stop screen sharing. Okay, did I stop? Oh no, I didn't stop. So screen share. Oh wait a minute, I'm messing this
Yeah,
up.
so this
There we
is
go. There we go. There we
so
go. Okay.
this is
Okay.
why it's a good idea to only screen share individual windows and not your entire screen.
Right.
It'll
Yeah. This is my first one, so I'm not good at this. How do
yeah, I
how
know.
do I give you the ability to screen share?
There should be a well, I mean, I can just do that, but you have to hit the present button. If
I got to
you
hit the what button?
you
Oh,
should be able to
you mean click? Just
present
click
me
it.
to all or something. Yeah.
Okay, I got you. Click. Okay, so while you're doing that, I'm gonna find this other site that says that is basically highly expressed and highly co-expressed too. And the
Sorry,
thing about,
I think you may have done the wrong one. Can you do the other
oh,
one?
you got the wrong one. Okay, hold on 1 second. Um, no, I can't. I'm sorry. You got two of them going. I got you. Okay, I see what you're saying. Okay, right. Okay, yeah, I've seen this before. And I tell you what, it kind of almost depends on the
Yeah, except
where you
so
go.
the really important thing about this is that if you see I've got this shown by isoform. So these are the different transcripts basically in different tissues. And if you see this red peak, right? So this is
I see that.
and the blue peak.
That's in the
These are the
test.
two trans.
Right.
Right. It's the peak is in the testes, but
Right.
the lines represent two different transcripts. And these two, the blue and the red, are the transcripts that don't cross the fusion site. The green one here, that's the one that fusion site. And if you can see, it's at 0 all the way across. Right.
Okay.
So no expression
Okay. So I got a
all
couple
the way
things to say
across.
about that one. I got a couple things to say about that one. You know, if it does, if it don't, you know, I don't. I'm be honest, I don't know a lot about that right there. If it does, if it don't, it doesn't matter. The fact is that gene is right there, and that gene is highly expressed, and you can't.
Wait, but hold on. I'm sorry. You can't say that though. You can't say
Okay,
that it's highly expressed when I'm
okay.
showing you
Well,
the
I
evidence that
got
it's not
different.
highly
Yeah,
expressed.
but you're showing me something, but I can show you information that it is highly expressed. But okay. I can tell you
But
what.
so
Let's say
the thing is that the source that you were just showing from ACE, right, is referring to all the transcripts. So if you look at all the transcripts together, you might get the impression that especially the red transcript that I was showing is moderately expressed. Now you didn't show any evidence that it's highly expressed. You
okay.
showed from
I'm gonna
ACE
show
that
you.
it's
Yeah,
moderately
I'm
expressed.
gonna show you some. But the thing about it is also this right here. You know, the thing about it is we are in our infancy of understanding genes, and especially understanding long coding RNA genes. You know why? Because we follow this evolutionary type of thinking that we're going to only focus on um, protein code genes that code for protein. So
So I'm glad that you brought
okay,
that up
hold on,
because I've actually already got open a another window. If you just keep
but wait a minute.
um,
You gotta let me finish saying what I was going to say though.
keep going. I'm just gonna
Okay, I
open
know.
up a
Yeah,
different thing though.
yeah. What I'm saying is a couple of things. One thing is long coding RNA genes don't have the higher expression rate as. Hold on,
Except they do.
no, they really don't.
What I've got open right now is a long non-coding
Yeah, but
RNA.
you can't point to the thing about it. You can't point to specific ones and say, okay, well this one do, so that means none of them do. It's not only that is you also have to take in consideration that there's also epigenetic responses there too. And how are you testing these epigenetic responses? How it's respond, and how are you testing how important it is when it's a network of genes? The thing is what the point I was trying to make to you is this whole area is highly networked. Okay, and the one thing is that's really important to say is that there are plenty with that DDX11L2 gene. There are plenty TSS transition start sites right there in that region where the polymerase 2 basically start its transcription right there. So you can't really necessarily go off of the very high transcription rates because of the fact is long coded RNA they transcribe lower.
Okay, so again, that's actually not true. If
Okay,
you look at any non-coding RNA that is known to have a biological function, it has a high expression rate. I just showed one example. This is XIST is known to have a biological function. It's non-coding RNA with a known function, and yes, indeed, high expression. If you look at any of the others that are known to have a function, it's the same thing. Um,
but the thing
so
about it is, see, I say that you guys kill me with that word function though, because that word function is so superficial. Because function, what does function mean? If it's serving a purpose, is it non-functional? I mean seriously, because really,
so so
because really, you guys got different meanings for that. Because if you ask Steve McCrae say functional gene mean if it codes for a protein. Same way he want to
so
call the same. Wait a minute, hold 1 second.
I would say
The
that
same way he want to call RNA long code RNA is junk and pseudogenes. But if they serving a vital purpose, they're still junk and they still not functional. So I mean, I just can't buy that.
well so I certainly would again I would say that the one that I was just showing is functional even though it doesn't code for protein. I would say that functional is something that affects the phenotype in some way. It doesn't have to be a direct effect, but it does need to in some way affect the phenotype. Um,
Okay, let me
otherwise
ask you a question. If you got a football team, you got a very successful football team, and you got a running back that's a running back who score points. He's going to score points for the team, and him scoring points is going to greatly allow the team to win. But guess what? What about that fullback that blocked so good for him? okay, that do the work, that do the work that don't get noticed, but it's very, very important. This is how genes work.
Yeah.
So,
Okay. So
hold on.
going.
Wow, man, come on, CRISPR, come on, man. I don't interrupt you when you're talking now. Genes work like that. Genes have very. God, baby, hold on for a second. You gonna kick me off the bed? Would you scoot over, honey?
Okay. Okay. So staying with your analogy, right? So DDX11L2 would be like a guy 3 states over not watching the game who happened to be wearing a jersey at the time, right? Like,
No, that's not it at all, partner. That's not it at all. Here, I'm gonna screen. I'm screen. I like that analogy. That's entertaining, though. That's sharp and that's witty, but it's not the truth. But let me go over to let me screen share something for you here. Is it this one right here? Yes. That's funny though. I'll give you that one. All right, let's see. Share. Wait a minute. So wait a minute. Application window. Okay. All right. Okay. So then I can just hit. Am I sharing? Am I sharing right now?
not yet, not yet.
Okay. All right. Yeah. So I'm not good at this right here, man. Okay. So I got share right here. Application window. That's what it's saying. Is it this? Okay. Am I sharing this right here or not? Okay. Wait a minute. Share.
There you go.
Okay. Okay. Am I? Okay. Okay. So I'm sharing this right here. Okay. So
Yep.
this site right here, Harmonize. This site right here, Harmonize. Let's go. Let's go about Harmonize real quickly. The reason I want to do about Harmonize. Oh man, wait a minute. Is that gonna work? The reason I want to go about that because I want to go about that because I want to talk about how a lot of these new data sets how they get information. They don't just get information the typical way that they used to. They use information from a lot of different data sites, and then they kind of combine the information and then the different resources to try to figure out exactly what these genes are doing. Because we, us trying to understand what genes, especially non-coding RNA genes, we're sloppy with that. It's hard to figure out what genes is doing. It's hard to figure them out. A lot of times you get an area of genes, you're not even sure what's going on because it's sometimes the area. So you have to really work hard to try to figure out what's going on. So this database basically it collects from 114 data sets and 66 online resources. It's a collaboration of data, rather it's structural or functional annotations, genomics, disease and phenotype associations, germline associations, protein sequence features. It has a more type of a cohesive way of finding what genes are actually doing. Okay, so this one right here, it lists all the associations and functions. It lists you got high association and you got low associations. High association, low associations. See, this is how
So you want to pick one of those at random, and we can talk about them.
No, I don't want to spend a hell of a lot of time on it, but I'm just saying, look, you got all this stuff right here.
So I've actually
This is a collaboration. This is a collaboration of all these different functions, all these different cell types is what he was talking about. But go ahead.
So I've actually gone through this website line by line, and none of this actually indicates actual function. And I can talk about that in detail on these. If you want to just pick one, we can talk about it.
No, look, see, I'm gonna be honest with you. I'm not gonna get into this non-functional thing with you because the thing about it is we don't know what's going on with that because of the fact is, we're just now hold on, real quickly. We're just now learning about long or we're just not really studying long coding RNAs. We haven't been looking at those. This gene you can't even, it's not even present in nothing else. So I'm not gonna spend a lot of time on, because I don't have a lot of time. I'm not gonna spend a lot of time going through all these different lines, whatever. What I'm telling you is what I said. There's a lot of associations here. Now you can knock down what I'm saying, but I'm not going line by line. Have you knock it down? But I will tell you that it has a lot of epigenetic responses too, like it has a histone modification, and then you also have in genes you have uh, where you turn off and on other genes. Okay, promoters. Now
Right.
these promoters may not register a high transcription, but that's pretty important to turn genes on and turn genes off.
Right,
When
but there's not actually any evidence that DDX11L2 does that, right?
yes, it does. Oh, yes, it does. They have
No,
promoter right
no,
there.
no,
It,
no.
it
So
has a
okay, so let's take that as an example, and let's go ahead and look at that line. Um, I, because the thing is, if you're gonna just say that there's evidence of function, but we don't know what it is, that I can't follow you there, right? Because if we don't know what the evidence of function is, then that means we don't have evidence of function.
no. You
So you
have
can say
evidence.
that you think,
No. I showed you
okay,
evidence right here. I showed you evidence right.
right? So I'm
I see anything
so I'm
about
saying
it.
that's
Okay,
so right. So,
I tell you what we need to do. We need to figure out what your definition of function means. What's
yeah,
your
sure,
definition of function?
sure. So something is functional if it affects the phenotype. Now, generally that means it's going to need to in some way affect protein level, although not
Uh,
necessarily. But so it doesn't have to be a protein itself, right? It doesn't need to be a protein itself, but generally it needs to affect a protein level at some point in some way. Now, there's a lot of ways you can do that. So obviously, yeah, if it's a protein, if it is coding for a protein, then that's pretty obvious how it's affecting protein levels, right? But a non-coding RNA could affect protein levels by affecting the transcription level of some other gene, right? For instance, or it could affect the histone modifications of some region, or it could do who knows what else, right? But it it's not enough to say that it's there, right? Or that even that it's transcribed, because that's not actually doing anything to anything else that's actually affecting anything. You have to actually show some connection between that that thing and the ultimate phenotype of the organism. That's what makes it functional. Is that decently clear, or is that if anybody's watching, we seem to be having some audio problems, so hopefully we'll get that resolved quickly.
Yeah. Can you hear me?
Yes, Cam.
Oh, so you should have said that a long time ago. I didn't know how to do that. Like I said, I don't know this stuff, man. All right, cool. All right, so okay, so now, so I'm gonna be honest with you, CRISPR. Seeing like you struggle, you were trying to be very careful with that function word. See, because you function typically, like you said, they associate that with a protein coding. So to me, function is something that's serving a purpose, something that's reliable and
Yeah.
viable. And
So,
according to encode, they use the word function a lot more flexible because the genome is important. It's important, to have all these things working together. The same way with co-expression, the same way when you have the genes is networked together. Are you trying to tell me that that whole network of genes
yeah.
that
So
work together in that fusion area, those genes are not important?
so what I'm saying is that you need to mute when you're not talking.
Wait a minute, are you muted?
Oh,
Oh,
I've got headphones.
I'm sorry. What you say?
I have headphones, but if you're not muted when you're not talking, then it echoes to me. So it's just sort of.
Okay, you want me?
Anyway, the way that I would define functionality is if it affects phenotype, right? So I was saying this, but you may not have heard me. There's a lot of ways that you can affect the phenotype. Most of those ways involve affecting your protein level, but not all of them. Um, but yeah, so it's not just it's not, you don't have to be a code for protein to be functional, certainly, but you do need to affect something at some point. Because if you're not affecting anything, um, I think that's fair, right? You'd agree with that at least, right?
Well, I mean, I think almost everything in a genome is affecting something. So, I mean, that doesn't really help your case. That helps my case more. That's being kind of generous to me because I consider things, like I said, a fullback don't score any points, but a fullback is important. If you have a gene that's getting epigenetic responses, his histone
Yeah. So
regulate.
if you're gonna, I'm sorry, but if you're gonna cite those lines from the Harmonized project, then it's important that
It's not
we
a harmonized.
understand.
It's not a harmonized project. That's see. The thing is we have to kind of we need to kind of bridge the gap as far as we can't evolutionists to me seem like they want to just go by that old code because that old code fits their type of thinking, calling DNA junk,
No,
calling.
have I firstly have I used the phrase junk DNA at all?
I ain't say you. I'm saying this is what evolutionists do, though.
But okay, but that's not going to be terribly helpful in a conversation with me, right? So,
Okay, well, but yeah, but you might not go that far,
but
but at the same time, though, just using the word function. Let's okay.
but can I just want to make a point, and it's really important. So is that if you're going to point to something, if you're going to say that, okay, so here I have this source, and it says encode histone modification site profiles, 195 histone profiles modifications near DDX11L2. Okay, so if you're going to say that, um, that's evidence that it's functional, we need to talk about what that actually means. Now, you didn't point to that one. I just picked one off the screen. But you did say something about there's histone modification profiles near it.
Yeah. Oh yeah.
So
Let me show you
right.
some.
So
I'm gonna show you some of the stuff that it's involved
sure,
in.
that's fine. But my point is just that if you want to say that that's evidence for functionality, we need to talk about what that actually means. And then if you say that once it's been explained what that means, that it's still evidence of functionality, that's fine. I'll disagree, but
Okay.
that's
Well,
my business, not yours.
I mean, we still gonna struggle with the word function. That's what's gonna happen. So I really don't want to.
So we can disagree. We can disagree with whether or not it means that it's functional, but I just want to explain what it means when it says that there are histone modification site profiles near DDX11L2. If you're going to cite,
So what do histone modifications mean to you? What does it mean to you when you have transitional start sites? When when the RNA polymerase is starting its transcription right there in that site, what does that mean to you? Does that
Okay,
sound is that important?
it could be. So do you know how long those sites are?
Hold on, what sites is you talking about the DDX? Hold on, what
No, the transcriptional start sites.
how long they are? I don't know how long they are, but if the RNA polymerase is starting there and it's starting its transcription there, I think that's pretty important there.
Well, it certainly could be, but that's not actually what I asked.
Okay. You asked me how long is it, so you tell me how long it is, and tell me this is the significance on the length. Because I have an idea that that you're going to say that it's not long enough to make a difference, or it's too long to make a difference, or whatever. So what was you about to say? Are you there?
Yeah, I'm just trying to find the
Okay.
yeah. So the actual binding site length is pretty small. Uh, I'm can't remember off the top of my head what the actual exact length is. It's like 10, 12 bases or something like that.
Oh yeah. I'm inclined to disagree with you on that one.
That's the length of the binding site.
I'm inclined to I'm inclined to disagree with you on that one. But I tell you what though, I want to show you this right here. Let's start screen sharing and let's go to application window. And I think it's this one right here, human chromosome. Okay, so are we looking at right here where it says they're talking about the binding right here? Do you see that? Additionally, they're talking. Wait a minute. Let me see. First of all, you realize that this gene basically it's reverse trans. It's transcribed on the minor strand. So basically, from the telomere to the centromere, right? The gene right here. Can you see this right here?
Oh, I'm sorry. You're not actually screen sharing.
Oh, I'm not screen sharing. Okay, hold on. All right. So let's do it again. So, oh, okay. Hit share, then do it. Okay. So let me see. So basically, this gene basically it's on the minor strand. So it's transcribing from the telomere to the centromere, in the other direction. Okay. So basically, um, from the 5' to the 3', reverse transcribe. Okay, so basically, this is what they're talking about this transcription factors here. Additionally, the function site contains data tracks for transcription factor binding 2A, 2B, indicating that it contains a functional DNA binding domain, specifically the three transcripting factors CTC, CTCF, CMYC, and POL2 has been shown to bind to putative fusion regions in the chromosome DNA sequencing. Okay, so basically,
So, well, hold on. Can you scroll back down for a second? I just
okay.
there's sort of a fundamental problem with that, right? So if you're going to say that the transcript has functional RNA motifs, right?
Right.
The RNA motifs can't do anything if the transcript isn't transcribed, right? So you have to actually have the transcript be present. Thing you would agree with that, right? Like if you don't have the transcript, it can't do anything.
But why you say the transcript ain't there?
Because there's no evidence of
What?
expression. There's no evidence of expression.
Now see, you know what? Hold on. See, using that one site, I use a site that said it was evidence that it was. You're using that.
No, wait, hold on.
Hold on.
I'm sorry, but you just said something that's wrong.
But you was referring to, but also you was referring to that one transcript. You didn't refer to the other transcript.
Yes.
Okay, so
yes.
so
So specifically.
Okay, so let's make this clear.
The website.
So let's make this clear. So are you saying that this gene is no good, or this gene?
So what I'm saying.
Hold on, no. Come on, let me finish real quickly. So basically, you're gonna throw this gene out. You're gonna throw the fact that is right there in the fusion site, that this gene is not important, because let's say you're right. Let's say that one transcript that right there on the site is no good. The gene is still there. Okay, so how do you have half a gene, okay, on one side of a chromosome and the other half on the other side of the chromosome if it goes across the fusion site? You can't have a gene on both sides of a chromosomes if they fuse together. You can't have that.
Well, you can't have it. You can't have it before they fuse together. After they fuse together, you can.
It's specific. So you trying to say that gene became, and that's the escape mechanism, but the gene is functional and it got functional.
No, it's not functional.
It's functional and it got, but the other transcripts are functional. But and I'm not.
No, they're not.
And I'm not agreeing with you by saying that the transcript is not functional. Okay, but see, what I'm saying is your case that you're building has a whole bunch of iffies. Okay, you got
the thing.
You got a
I don't need.
You got a DDX gene that's transcribing and on the minor strand that has transcriptional start binding sites there. You have epigenetic responses, whether it's methylation right here, and it says it all right here. Here's all.
Actually, so again, you don't have you don't have methylation in response DDX11L2. You have methylation near the region of DDX11L2, and that's a huge difference.
No.
That's that's true.
But see, this right here support. Okay, I tell you what, this supports what it said on that site. It support what it said on ACE when ACE said it was moderately expressed.
Right.
It also supports what the ENCODE project has been saying.
Right. So I think that it's really important that we get something across here. So you go to that ACE website, right? And it says the DDX11L2 is moderately expressed, but they're referring to all transcripts. Now, when you then go to this paper and he starts talking about the one that goes across the fusion site, that's only one transcript of several, right? So the one transcript that goes across the fusion site is not moderately expressed. It's at all, basically, right? So you can't conflate the two. You can't go from one transcript that doesn't cross the fusion site is moderately expressed. Therefore, the other transcript that does cross the fusion site, it doesn't work that way.
So also you want to maintain that it doesn't have function, okay? But at the same time,
well, that's a different question.
Hold on, and plus you also can't plus you that, like I said, that gene isn't in mouse. That gene isn't in chimp. So you can't knock that
irrelevant
to see exactly what that gene is doing. So you can't say anything for sure. You can't say anything with a high degree of certainty. You can't say
right.
You can't say that just the mere fact that this gene has all these epigenetic. You can't say that this gene is not getting epigenetic responses. From histone markers. You can't say that because go ahead.
Go ahead. Okay, so saying that there's no evidence that it doesn't have function is not evidence that it does have function. First. Second, saying that possibly transcription is affected by other things is not evidence that it affects the transcription of other things. Okay?
Okay, I'm listening. Go ahead. Well, I tell you
so
what.
So in other words,
here. Okay. Okay, so which browser did you go to? The US the UCSC genome browser? Because if you can look at what I'm talking about, he's talking about these transcripts right here, variable and complex. He's talking about you got that second promoter and the first intron. That is actually the fusion site right there. That second promoter and the first intron. There's no fusion there. Okay. Then you got, and then you have, and then you have a gene that's on both sides of a fusion site. You got to deal with that. Then you want to say that this gene don't have any function, but yet you can't really test the gene for its function because this gene is not present in
Okay, so
any.
I don't. I'm sorry, but the burden of proof is not on me to prove that,
Well,
even though I think I have, it is not on me to prove that this thing doesn't have function. You need to demonstrate that it does have function.
okay, well, I tell you what, I can demonstrate that it got function because of the fact that it has transcriptional binding starting sites.
Doesn't demonstrate function.
But you gotta let me. But even if you don't like what I'm saying, you gotta let me finish. Okay, I can say that, and I can say that it's highly co-expressed. Okay, and I can also say that it's highly networked with a whole bunch of other genes. I can also say that that it's affected in more than 255 tissue types. I can say all that. Okay,
And none of that is evidence of function.
and I can say that it spans the fusion site. I can say all that.
Right, and I can, if you want, I can talk about each and every one of those things and explain why that's not actually evidence of function,
Well,
but you don't want to get into that.
You
So
already said that. Okay, so let's move on a little bit here. Let's move on. So
I mean, I don't want people to think that I'm just bluffing on this. I mean, if you want, we can talk about each and every one of those things.
I can't listen. I got a girl here. I can't talk for a long time. I was telling you that, dude. So let's move on. Let's move on to let's move off of transcription and let's move to other stuff here. I'm trying to see what else we have. Yeah, because okay, so talk. Well, actually, talk to me about this website right here. Are you? Am I still screen sharing?
Yeah, sure.
Okay, so then we have this right here. Okay, we got it says DDX02 is highly expressed and co-expressed with DDX11, which is, I think DDX11 is a protein gene. Am I not correct there? That's a protein gene, right? The
I'm not
DDX.
sure. I have to look it up.
No. I'm sorry. No. That's a. I don't think that is. But the thing is here is that whole fusion area. You have over 16 genes, and I think it's 16 genes and 24 um 24 pseudogenes there, or vice versa. That whole area is a super functional area. So what you're trying to tell me is two chromosomes came together in a highly functional area, okay, right there on the ends, and just can't you slam two chromosomes together, and then you got all that functionality after you had all that damage. My thinking is this here. Hold on, let me check one thing. My thinking is this here. If you had damage up in the subtelomere regions of that gene, I think you would have killed a bunch of those binding sites. You wouldn't have had significant binding sites right there. You would have had a serious problem.
Okay, so firstly, I would dispute that it's a highly functional region. Secondly, you have to understand how small and common binding sites are. I once went to a binding site search engine, and I submitted a random string. It was about 1,000 bases long, just completely random, and it found something like some binding sites on this completely random string. So it's not enough to say that there are binding sites. That's just not evidence of function to just to just say that there are
That's why,
binding
that's why I
sites.
say you killing me with that word function, man. You killing me with that word.
Okay, so what does it do?
I'll be
What do
honest
you
with you.
have evidence that it
I'm gonna be honest
actually
with you. We
does?
don't know. Listen, we don't know what law.
If you
We are
if
learning.
you don't know
Listen, right
what it
now we are
does,
learning. Hold on 1 second. Now we just started looking at non-coding RNA genes. They're associating long-code RNA genes with disease now. Okay, we just started learning about. We just started learning about dual coding genes, duons. The epigenetics is relatively new to us. We don't know none of this stuff because you know why? We were so fixated on protein coding genes, so fixated on them that we're just now learning stuff. And evolutionists don't want us to know this stuff because evolutionists want to believe. This is what they want to believe. They want to believe that all this other stuff is just remnants of past species, just evolutionary remnants. That's all. That's what they want to believe. So I just don't buy that. The whole function, the whole function thing, what you guys talk about, I just don't buy it because if it's serving a vital purpose, to me, that's function. So
but you don't know that it's serving a vital function. The thing that you don't know that it's serving a vital
the whole
purpose. What you just
the
said
whole
was
genome is important. The whole genome
no,
is
but hold on. I'm sorry. But you just said what it
oh,
does. If you don't know what it does, that means you don't know if it does anything.
uh,
Okay, you can't say that we don't know what it does, therefore it's functional. That doesn't
I
make any
know
sense.
I know I know it's in a highly functional area. It's highly networked with other
It's
things.
not a
Okay,
highly functional area.
okay, I tell you what. Let me ask you a question. If Tompkins' work is so sloppy, okay, what he's doing, how come there's no one beside you YouTube guys trying to refute him? His work is
Uh,
relative. His work is published to where anybody can get their hands onto it and do something with it. Fairbanks started off trying to go against him. He went quiet. What's his name? Kenneth Miller. What did he do? He pushed his whole thing. He went in silence. Why is it no one beside YouTube people trying to refute Tompkins? Why?
because people who have actual labs like to do things that actually matter.
No. You got
Right.
no,
So this is
you
a
got
this is a I'm sorry, but this is a decided issue. Okay, this is done as far as
it's not the same.
anybody with actual training in the field is concerned. People who are have actually been studying this, who've actually been working in the area, they don't see this as an interesting issue worthy of research or worthy of publication because it's been
That's
done
not
to
true.
death.
That's listen. That's not true at all. Any research that's been done on that has been years and years ago. Are you telling me that with the whole idea of creation versus evolution, we got huge debates of scientists that come on TV debate that stuff? You trying to tell me that something that's significant as the common ancestor to human is not important? You got this guy came in here and wiped all that mess out, who changed the whole idea of that. He basically, because you got so many different things that can't happen, with this fusion. You can't start a species off with a low viability to produce offsprings and expect to be the dominant species. You
Well,
can't have, hold on for 1 second. You can't have a fusion that's so degraded to a point to where you only have a base of 800 when you used to have 30,000 on there. You can't have two centromeres on a chromosome, and not knowing how that centromere actually got deactivated. And then you can't, and then even those alpha 8 sequences that you see, them alpha 8 sequences isn't specific to any alpha sequence that you see all over chromosomes. You got so many question marks about this. You trying to tell me that Tompkins didn't deliver a gang of question marks? The stuff that people already knew. As a matter of fact, if there wasn't any question marks on it, they wouldn't been trying to have escape mechanisms, making up reasons why they're trying to figure out why the telomeres basically lost all those sequences. They wouldn't have escape mechanisms for it. So
look, I don't know.
You can't tell me this is a done deal, because it really isn't at all.
Look, firstly, I don't really know what to say about this escape mechanism thing, because if you're just going to point to any explanation of how things actually happened as it being an escape mechanism, then this conversation's not really going to go anywhere.
Okay,
right?
wait a minute. But are you telling me so you definitely telling me that what we were saying about those telomeres fusing, that is definitely the reason why there's definitely no chimp DNA on human chromosome 2, and you got all these copies, these 18 copies of this this RNA helicase family, that the DNA is definitely gone, and it definitely diverged to just specific human, and that all that DNA is just gone because it's just gone, but with no mechanism on how it left, with no mechanism on how telomeres that never fused before in history can fuse to that degree.
Except they did, but yeah.
I mean, I guess this won't go anywhere because we had a stalemate because right now you're not being honest.
Well, I'm sorry, but there's evidence of fusion events in other animals in several different species. There's evidence of head-to-head fusions.
But that's satellite DNA to telomere.
No, it's telomere to telomere. It is head-to-head fusions. I'm sorry that it is there. In species of pigs, Hartmann zebras, yes, they survive.
Yeah. I heard about one that might be in a pig, a possible. That was it. I heard something about ants,
but zebras,
but the whole
and
thing,
ants,
and
fish,
that's speculation.
And antelopes.
Those are pure speculation.
And rats.
Nobody's talking about that. That's pure. Those are pure speculation. Period. There's no real evidence.
Well, except that we've got the fish staining, we've got the sequence data, we've got
but the fish staining that they use support the fact is that there is that chimp DNA that's lost. How did you lose the chimp DNA off of chromosome 2A and 2B? How did you lose that stuff? Where did it go?
Look, there are several mechanisms of large scale deletions that are possible.
Wow.
I don't know how much time you really want to devote to that, but especially when you're talking about regions where there's a high degree of internal similarity, you tend to get folding events where they sort of there's non-homologous enjoining. Basically, there's a break, and then it kind of loops over itself and reattach and gets ligated to itself. Again, I don't know how much time you want to spend on that, but that's fairly well understood.
I will send you this thing right here, just on the side chat. This guy basically talked about. Oh, wait a minute. He talked about how there is almost, if you look at the genes that's on there, there's hardly any matches. You're saying that those genes, there's a lot of matches. That's
I'm sorry. I think I missed what you just said. If you look at which genes that are on there, there's no matches.
I'm sorry, what?
So you just said
what did you just say right now?
You just said that if you look at the genes that are on there, there are hardly any matches.
Chromosome 2. We're talking about chromosome 2.
Okay, yes. So if you go from the top to bottom of chromosome 2 and compare it to chromosome 2A and 2B on chimps, you see that the synteny runs the entire length of the chromosome, right? With matches on proteins on both sides and non-coding regions on both sides, all the way down. The synteny.
This guy looked at him
exquisitely accurate.
And he went to let me see. He went to you can look at that later. I don't want to. I don't want. That's a 5-minute, 4-minute video. I don't want to do that right now. But I mean, I just we're not going to agree on this here, partner, because like I say, there's way too many. To me, there's way too many like huge questions. And for one serious one is, how did it get so degraded within that time span? Where's all that DNA that's supposed to be there that's not there? You know, I don't see how how there's a fusion there. But you don't have if you don't have the evidence of the DNA there, if you don't have the DDX LL2 gene nowhere near on chimp, nowhere near on none of the chromosomes, you can't say it came from another chromosome to jump. You can't say that because it's not there. You got 18 genes in that family, only 2 of them present in chimp, and none of them present on that chromosome 2. Where'd that information go? Are you there?
Yeah, just 1 second. I'm just trying to find something here.
So you don't.
Wait, no, actually, I already did find it. I forgot that this is using the old terminology. So yeah, actually, you have a copy of DDX11 on chromosome. So it's there.
Where you have it? Where at? He did the fish analysis. It's not on there. He did the fish analysis.
Well, if who did the fish analysis?
Hold on. What's the name? It was Ventura was perplexed. I think it was Ventura, or was it I gotta find out. Wait a minute. Okay, here we go right here. Let me start screen share this for you right here. Let me see right here. Okay, so let's go to screen share and let's go to application window. I believe it's human chromosome 2 right here. Costa. Well, Costa postulated that he proposed variants of the DDX11 gene had evolved from an ancestral sequence of apes. However, when the DDX gene sequence was used in the fish, the fluorescent in-situ hybridization, it only hybridizes in 2 places on chimp chromosomes 12 and 20. Okay, and then the same experiment was done for gorilla, and it basically showed synteny on chromosomes 3, 6, 7, and 20, which is basically a complete contradiction. What it's saying here showed no overall synteny of 2A and 2B. Then they talked about how much specific chimp DNA wasn't there, how much specific human DNA was there. 18 copies of the DDX gene, only 2 copies in chimps and 4 in gorilla. That's a serious that's a serious statement right there. You know, you can say stuff like you can have an escape mechanism and say, well, they diverged into something else, or they got lost some kind of way or whatever, but it doesn't make a lot of sense, especially over that time frame that you would have the divergence from chimp to human, that you would lose all that information. You would lose all those telomeres, you would lose all that, the 24 megabytes of information, and then the whole thing about dealing with the 2 centromeres. You don't really have a good plan on how you deactivate the centromeres. Because you got this thing called a fusion bridge that happens, and that's cancerous, where you have 2 centromeres on there.
That doesn't happen in zygotes, so that's not really relevant.
It doesn't happen in what?
So that's something that happens in that doesn't happen in germinal cells.
It happens when you got a telomere. It happens when you get 2 telomeres that fuse together, partner, because you got 2 centromeres.
Sorry. It happens. It happens in the case of somatic cells when the two sister chromatids are fused together, right?
Right,
And
okay,
so
yeah,
so
right,
they
yeah,
need to get pulled apart to go in different directions on the cell.
right.
If they're already fused together, though, right? If they're already fused together, that's not the way they get pulled apart. So it just does not happen
Yeah,
in that circumstance.
but two centromeres is going to, you're going to have all kinds of issues with two centromeres. And how do you deactivate a centromere? How does it
One second.
deactivate itself? Do you know?
Sorry, just one second. So yeah, there's actually been a lot of research into that.
And nobody came
I
up
think
with nothing real solid. I can tell you that right now.
well, I mean, actually, there's a paper from 2013 about epigenetic. So that could be a decent answer. I haven't read this paper, so I don't really know.
No. I'm talking about centromeres deactivating itself. You know, you can
Yeah,
deactivate a centromere. You can do it in a lab. You can do that in a lab.
no. That's what this is. This I again I have not read this paper yet, so I don't know. But
Oh,
just quickly scanning through the abstract, that is my impression of what this is talking about. Again, I'd have to actually read it to be sure,
right.
but
Yeah, you don't sound too confident with that. But what I'm saying to you is there is okay. Put it this way here. Uh, let's try to wrap this up and close. Okay. Put it this way here. Okay. Let's say I'm pretty sure I pointed out a lot of bad things that you can't account for at all. You can't account for some of this stuff. You can't account for the missing. You can't account for the chimp DNA that's not missing. That's missing. You can't account for that two chromosomes that got fused together, and then you don't have those genes on there that's supposed to be there. You can't account for the fact is that you have so much degradation of those telomeres within that time frame when telomeres are built not to fuse. You ain't giving me no good examples of no telomere to telomere fusions. Okay, the only thing that's near that was the muntjac, which had a different type of fusion. Okay, so those are some serious problems. And then the whole thing about the centromere. What happened to that centromere? Those are some serious problems. Then the whole thing about the humans becoming the dominant species, and they basically don't even bear good, strong offsprings. So it's just a lot to deal with, man.
yeah. So like I can try and hit those. I probably won't remember all of them. Again, so the viability of so offspring are either viable or they're not when it comes to this, right? So it's not that the offspring are going to live, but they're going to be weak. It's either spontaneous abortion, right, immediately, or normal offspring, right? So it's okay. So it's sort of inconvenient, but realistically, it's not really a huge detriment.
Yeah, that's a
Seems
huge
like.
detriment.
So I'm sorry. What part of that do you think is a huge detriment?
Because you're not going to be able to become a dominant species if only 2 out of 6 of your offspring survive. And I don't think
No. So
in some
okay,
cases
wait, hold on. No, so
make
I'm sorry. That's a misunderstanding. That's a misunderstanding. I'm sorry. It's not that 2 out of 6 of your offspring survive. It's that 2 out of 6 fertilized embryos survive. So in other words, 4 times out of 6, when sperm meets egg, right? Spontaneous abortion. That pregnancy does not last. That means that the mother can, the couple can try again right away. Okay, so which, let's face it, they're going to be trying again anyway, right? Because
right.
I mean, we know how these.
I'm thinking.
Right,
I'm thinking.
you know what I mean, right?
I'm thinking there probably might be some birth defects involved there too.
No,
It might.
there's not birth defects. That's the point. Is that
I gotta look. I gotta look into that one. But
right? So when you have that 2 out of 6 where it's the right genetic complement, that is a viable fetus. And when I say viable, I mean it's normal.
But see, you know, the thing is, when I talk to evolutionists about this stuff, man, you guys need a lot of help. You guys need a lot of help to make things work for you. It's like say, for example, when we talk about mutations, I was talking to Isaiah, and we was talking about how, he wants to say that they that bacteria was once bacteria, now it's our mitochondria. That bacteria got comfortable in the eukaryotic cells, and then some kind of way from there, the cells that Yusuf didn't have a nucleus, formed a nucleus. Then you got the mitochondria that came from bacteria. Then you got these organelles that evolve, and then you got all this machinery that evolves. And it's like, where is the information that made this evolution happen? Where is the mechanism that made this stuff happen? But we believe it because we believe in evolution, but we don't know how it happened.
Hey, quick question. Are you familiar with chlamydia?
Chlamydia?
Yeah.
Yeah, of course, that disease.
Yeah. Do you know what the bacteria that actually causes it? Do you know anything about it?
Nope.
It's an obligate intracellular parasite. Okay, that means that it lives inside of cells.
Okay, so what I'm saying to you, bacteria stay bacteria. Okay.
Right. So the mitochondria.
Bacteria can't evolve into a mitochondria and be the power source for cells. Cells do
actually.
Cells. Wait a minute. Let me say one thing real quickly. Cells do not increase in complexity and change from not having a nucleus to having a nucleus to not having the machinery in it to having machinery in it, because the actual proteins that's the machinery is made of is the actual DNA information that's necessary to build it. So those two things have to be made at the same time. You can't get the cell without the DNA. It's a chicken and egg issue. You can't evolve and increase information from a bacteria to a mitochondrium when bacteria only stay bacteria.
Well, actually, mitochondria are less complex than most bacteria.
The bacteria. Oh, okay. I'm sorry. Yeah, bacteria is very complex, but bacteria stay.
Mitochondria have actually lost most of the stuff.
I'm sorry, but mitochondria is what powers the.
Right.
I don't know.
The thing is, actually, there's a lot of people. There's a lot of people who.
It powers the.
Yeah, no, I know mitochondria. So I'm just trying to make a point. There's a lot of people though.
But
just really quick.
Roles like that. You gonna swap?
There's a lot of people
to that.
There's a lot of people who actually would argue that mitochondria should still be classified as bacteria because they have their own independent DNA. They replicate independently, sort of independently. Right, so that's an argument.
Homology don't mean ancestry. That's the whole thing. Homology could mean common designer. That's the whole thing. And people who argue that are just straight out evolutionists who wants to believe the evolutionary paradigm no matter what.
Right, but I could say that people who argue for the common designer are people who just want to believe in a common designer no matter what. It's not a helpful argument.
Okay, but the thing about it is this here. We know how things increase with information. We know how things get complex. Okay, and we know it's a building process. We know. We may not want to believe, but we know for a fact that there's just no way in the world you're going to get from single cellular up to a complex brain by natural processes. It's not going to happen.
Well, that's an assertion. That's not.
What's the mechanism to get there?
Okay, so you understand that if you make a statement that it's impossible to get from A to B, I don't have to give the mechanism to get from A to B. You have to provide evidence that it's actually impossible to get from A to B.
No, wait a minute. That you know, that's how. See, that's just like
You made the claim. No, I'm sorry.
no,
You made
hold on
the
a second.
claim. I
Hold on, no, because the point is obvious and clear. That's just no. You can see that's just like the whole thing about where you know I got all this different empirical evidence that the DDX1 has all type of function, but since I can't say specifically exactly what it does, exactly what it does, what the function is, that means that I'm automatically wrong. But yet
well,
you can
actually,
tell
no,
me,
actually, that's
but
different.
hold on,
That's
but you can tell me that single cell, even before the single cell, the nucleotides in a primordial ocean, okay, basically some kind of way sequence themselves specifically to make the first cell. And some kind of way that cell made the first simple cell organism. And through some type of mutations that it increased information all the way up when we know that it can't do that because you need the DNA information to do that. And we know it's
okay.
not
Again,
present there. We
that's an
know that.
assertion.
We know it can't happen.
No, no,
No, okay,
no.
we know.
You just asserted it can't happen.
No,
You
no,
can't just assert it can't
we
happen.
understand. We understand how DNA work. We understand. We understand that in order to build proteins, you need DNA information to be specific. Okay, and we know that natural processes can't do that. We know
Hold
that.
on, I'm sorry. Are you saying that RNA can't be functional? Because I thought earlier you were saying that RNA could be functional.
Okay, but the problem is RNA has to be sequenced too, and plus RNA really don't live outside of the body. RNA RNA is inside of the body. The reason why you want to say that RNA can do it because RNA has I'm getting tired. Catabolic abilities, but it doesn't matter. RNA can't increase the information to do that.
Actually,
That
just
information
really
is
quickly, I wanted to point out
infinite.
that there actually are varieties of naked RNA virus. So you do
Okay, but
actually find.
a virus that ain't necessarily considered a living. See what you're doing is you're jumping over mountains with nothing, and this is what evolutionists do. You point these little small things, and then you say, okay, well, because we got that, then we can get this. That's a logical fallacy. It's not true.
I, I didn't say you could get it. I was just responding.
Well, you was making
You
a point
said,
that
I'm sorry, just
okay,
let me finish. You said that you don't have RNA outside the body. I was making a point that you do actually have naked RNA viruses. So you do have RNA outside the body. I was responding to a specific
no, I
point that
thought
you
you said
raised.
okay, no, but you said RNA something. I forgot. Okay, well, yeah, but see, viruses isn't even considered living because viruses they need a host. They can't do anything without a host.
But
They
how is
can't
that?
do anything without a host. So
I'm sorry, but
they're not.
how is that?
But wait a minute. Are you saying viruses is considered living? Is that what you're saying?
I I would not personally classify
Okay,
them
they're
as
not
living. They're
exactly. They're not considered.
virologists would, but that's them. Their virologists tend to be weird.
Well, Crispr, I think I'm gonna close this, man. I'm getting tired, man. But it was nice. It was civil. It was cordial, you know. So, you know.
Yeah, hey, and you know what? If you ever have time, and you want to go through one or two of those lines from the Harmony Project, I will go through them with you and explain why they do not actually imply function in any way.
But the only see the only reason why I'm not really game for that is because of the fact, like I said, we don't understand long coding RNAs. We don't understand.
I know, but so
We
I'm
don't
sorry, but this
understand.
is the thing,
We
right?
don't
So
understand.
you presented.
We don't understand gene expression all like that. We don't
So
understand.
that's
We are learning so much about the genome. It's ridiculous. It's
that is a perfectly hold on, that let me just that is a perfectly fair argument for why it might be functional, but you're trying to say that it is functional, and to support that, you're pointing to the Harmony Project, right?
It's not, but
is
it's not
you.
a project. It's a website. The
I know, but
encode is a project, and the encode is a project of over 800 scientists.
yeah. So the thing is, my point is that you can't point to that site and say this is evidence of function, and then say, well, it doesn't matter if that's not actually evidence of function because it might have function,
No, that's not what
right?
I'm
Because
saying.
that is what you're doing. That's exactly what you're doing.
No,
That's
no, no.
exactly what you're
What I'm
doing.
saying is for one thing is, like I said, we got different. The word function is designed for evolutionists to be able to have something to hold back from creationists.
What's your
Because
definition of function?
again,
Is it different from the one I've been using?
my definition of function means something that serves a purpose, something that serves a purpose in the genome. That's function to me. It's
Is that
it's serving a
is
purpose.
that is that different in any relevant way from something that affects the phenotype?
No, I mean, if it's serving a purpose, it's serving. If it's necessary and it's needed, it's serving a purpose. It's functional. You know.
Okay, so just let me see if I can't provide a counterexample, and you just something that isn't functional. If you can imagine uh, uh, just series of mutations, just like completely randomly in the middle of a genome, right? And you get like 50 bases added in, just completely in the middle, completely random. Not functional, right? Doesn't do anything, just sits there. Not functional. You would agree.
Okay, if it's okay, now I agree with you. If it's just sitting there not doing nothing, yeah, that's probably not
Right.
considered function.
Okay, so because
But
the thing is,
you have to establish the fact that that's what it's doing, because it. Okay,
when
go
you
ahead.
have a definition, like this, sometimes you have to define it by what it isn't. Okay, so I'm just trying to figure out what can we say isn't functional, and
Right.
then everything else is functional.
If it's absolutely not doing anything, then I would say it's definitely not functional.
Right. So here's a question. If you have something, and other things are doing something to it, does it have a function? Right. So if
Yeah,
B
because
is
it's a vehicle. It's being used as a vehicle for something. Yeah, of course it's a function. It's functional.
no, I'm not saying it's not a vehicle for anything, right? So I'm saying if A has a function, right, of messing with the histones around B, and it happens to also histones around C, okay, and we know what B does. B does something with the phenotype. Who cares what, right? Does C have a function necessarily?
Well, I mean, the thing about it is that's a little bit vague. And the thing is you probably wouldn't know if C had a function. You probably wouldn't know.
Right, exactly. You wouldn't know because it doesn't say that it's.
So you can't say that it's just bouncing around C because you don't know if it's just bouncing around C. That's the whole thing.
No, so the important word is just, right? So all you know is that histones get modified around C. Who knows if C does anything? The fact that the histone gets modified isn't evidence that C does something. It's evidence that the histone maybe does something, and it's certainly evidence that A does something, but it's not evidence that C does anything. It might not, who knows?
Well, I'll put it this way here. You know, even if you want to say that one transcript is not functional or whatever way you want to say, it has more than one transcript. Okay, and the fact
Yeah, but only one of them crosses the fusion site, so that's the only one that's relevant to this
discussion. It doesn't matter. It doesn't, but that gene is there though. That's the whole point.
I know, but
the whole point is the gene is there.
No, I'm sorry. That's actually not the whole point. The whole point that you're trying to make is that if this gene crosses the fusion site, that's a problem, right? So
the whole gene crosses.
Only the one transcript does. The other transcripts don't cross the fusion site.
Well, okay. Well, I doubt it. I'm gonna look into that though. But it's the fact is that whole region it's extremely functional. Okay, the whole region
So
is extremely
that's
functional, and you don't.
that's an
This
assertion that I
and
disagree with.
you don't get the type of functionality by slamming two damn telomeres together. You don't get the type of functionality. It doesn't make any
Yeah,
sense. If it's already
I
functional,
agree. I agree. I just reject the claim that
you
it's
think
functional.
you think the whole region of all those genes is not functional? All those the 16 genes, the mRNA genes, and also the two genes there. You don't think those genes are functional in that whole region? You really don't think them genes is functional? You think that
Um,
that
some of them might be functional. I would have to look into it specifically, but the question really becomes, um,
well, that's where you
if they
messed
were
up.
if they are functional, would that function necessarily have been disrupted by the fusion? And if they're in the subtelomeric region, there's no reason to think that the function would be disrupted by the fusion. So what?
That you're not going to get you're not going to for one thing um you, you're not going to get two chromosomes coming together, okay, and then you're going to get functionality right there, and especially when
Hold
you
on,
got
wait,
all
hold on.
this damage, you got all this damage from, like I said, a maximum of 30,000 to 800 bases.
In
and
the
then
telomere.
and then well, I mean, you guys need to get together and figure out how you guys want to play that, because some people play it differently. Some people say subtelomeres, some people say it's in the telomeres. You know, but what you guys want to agree on is you guys want to agree on the fact that the damage stopped right there near the binding sites or what have you, where nothing got damaged there, right? But yet at the same time, you're missing a gang of chimp DNA. You're also
Yeah,
missing you're also missing a gang of rich chimp. I can't remember what I was trying to say right now. Look, I got to get out of here. I'm tired, man.
she's gonna kill you, man. You better.
Yeah, I know she is. But it was pleasant talking to you, though. I appreciate that. That's
Yeah,
the reason
it's a
why
good talk.
That's the reason why I decided to talk to you instead of talking to Isaiah, man. We would have been arguing with each other too much. I didn't want to do that. So
Yeah, I think this was a good amount of arguing with each other.
yeah, but it wasn't. It was civil. It was civil.
Right, right. So
So definitely
try to do it.
definitely disagree. But
Yeah,
yeah, you have a good one, man. Who was the viewers?
sure.
It said 3 viewers. Who were they?
I have no idea. I was one of them, I'm sure, because I have it open in a
Okay,
different
right,
tab.
right, right, right. Okay, okay. So is this thing gonna be? I don't know if it got recorded automatically on my end. Is it recorded
It
on your end?
it's as soon as you end the call, it should start uploading to your channel.
Okay. As soon as I hit stop broadcast.
Yes, that's right.
Okay, okay. Well, that's what we're gonna let it do then. But look, hey, nice talking with you, man. We talk about something. That
Sure.
um, I actually want to talk to what's the name though. I like General Solo. What's his name? General Han Solo.
Yeah, yeah. He's a good guy.
I disagree with him on the age of the Earth, though. I'm all the way young Earth. I disagree with some of the things he
Well,
say, but
that's probably not a good thing, but yeah.
right. Well, yeah, but I don't mind going against the fray. But um, I will say if evolution happened, it would happen the way he says it, not the way you guys think it. Because evolution would need some help. What do you think about that?
Well, I mean, I think that it's fairly natural that if you believe in God, that you're going to believe that God was involved in the progression of life. I mean, uh, that that seems to follow. I mean, no quarrel with General or Fiona or Jade or Wayne or any of the others with that. It's a
Right. Well, they would.
natural position.
Right. So you absolutely believe there's absolutely no God. How certain are you that there's no God?
I don't take a position on that. That's not my issue.
Oh, you just believe evolution happened, basically.
Yeah, I'm here for the science. I get into philosophy occasionally, but not that much.
Okay then. Okay, Crispr, you have a good one, man. I'm gonna go
Yeah,
ahead and
sure,
end
you too.
this. Take
All
care.
right, it's good talk. Yeah,
Yep.
see ya.